This has been explained by a particular theory of chemiosmosis or chemiosmotic theory by Peter Michell. He is a Nobel laureate for this particular theory. What chemiosmotic theory explains is that as the protons are accumulated inside thylakoid luminal space, a transmembrane proton gradient of about 3.4 pH units is created. It's because the thylakoid lumen will have a pH of around 4.0, while the stroma will remain at around 7.4. So there will be a proton gradient of about 3.4 pH units. This creates a proton-motive force and an electrochemical gradient, which is responsible. That energy is responsible for the synthesis of ATP, and this H plus can escape back to stroma through ATP synthase alone. And the membrane is impermeable to H plus ion, so it can escape through this ATP synthase alone. And the ATP synthase of mitochondria as well as chloroplast are shown here. They are structurally quite similar. We have actually explained the structure of CF1-CFo complex in oxidative phosphorylation, and this CF1-CFo complex is quite similar to it in structure. It has been conserved for billions of years throughout the period of evolution. It's been conserved structurally and functionally. And this is called CF1-CFo complex because C represents chloroplast, and the structure is the head is similar. You can see 3 alpha, 3 beta, gamma, delta, epsilon subunits in the head, or F1 part, and FO part, the stalk, is having A, B, B dash, and C subunits, which forms a ring. So we can see the moving unit rotor as well as the stationary unit stator also here. It is quite similar. The structures are similar. We can see what are forming rotor and what are the subunits that form stator also. So this is ATP synthase, and we can see here the H plus accumulation. It is different. Actually, the process is a bit different from what occurs during oxidative phosphorylation. Here inside the lumen, H plus escapes through this ATP synthase so as to form ATP molecules. So this is chemiosmotic theory. The structure have been shown here, explained here, which we have already discussed. You can go through. And chemiosmotic theory is also explained here. And one of the things that we have to note here is that a pH gradient of 3.4 is essential. And if the gradient goes below 2.0, which means the accumulation of H plus has to be perfect. If the concentration is less, H plus accumulation is less, to create a gradient of less than 2.0 only, then ATP synthesis will not occur. So this is chemiosmotic theory. You can go through it also. Okay, so this figure further illustrates the process of chemiosmotic theory and synthesis of ATP. You can see here the hydrolysis of water creates H plus ions here. Then cytochrome BF complex again contributes to the concentration or accumulation of H plus inside thylakoid space, as well as the H plus that is taken away from stromal space also contributes to the difference in H plus ion concentration across this membrane. That means the stromal space and the thylakoid space, across that membrane a proton or electrochemical gradient is maintained. As H plus accumulates here, it will escape through ATP synthase alone, because this membrane is impermeable to H plus ions. It can escape through this system only, ATP synthase, CF1-CFo complex. So this energy is used actually to synthesize ATP from ADP and inorganic phosphate. So this theory, chemiosmotic theory, has been experimentally proved also. That was in 1966 by a person named Ander Jagendorf. What he did was that chloroplasts were maintained at a normal pH of pH 7, and it was suddenly taken into a solution having pH 4. Now the chloroplast acquires pH 4 and the solution, the outside solution in which chloroplast with the pH 4 inside it is there. The solution, the pH of the solution suddenly changed to pH 8 along with addition of ADP and inorganic phosphate. Now, a burst of ATP synthesis happens together with disappearance of the pH gradient, so across the thylakoid membrane. So this was observed, and this was a very good experimental proof for chemiosmotic theory. And we can compare the process of synthesis of ATP that take place during photosynthesis with that of oxidative phosphorylation. The photosynthesis involve chloroplast when oxidative phosphorylation take place in mitochondria. The protein cluster assemblies are similar but structurally different also. The assemblies are arranged on the inner membrane in mitochondria, whereas it is arranged on the thylakoid membrane in chloroplasts. The accumulation of H plus take place in lumen, whereas the accumulation takes place in mitochondria inside intermembrane space. The involvement of ATP synthase is similar, the structure also very similar, and the theory behind H plus and getting converted or used up for synthesis of ATP as chemiosmotic theory is the same followed during both the processes for production of ATP. Now the summary of light reaction is overall input is light energy and water. The overall output are ATP, NADPH and oxygen. And the basic strategy followed by plants was that the ATP and NADPH are produced during light reaction, then they are used up during dark reaction.